Today's lecture is about red blood cells. We will discuss about the structure of these cells, variations in the size, shape, number, and the functions. So first of all, let us understand what are red blood cells. These are also called as red blood corpuscles, or these can be abbreviated as RBCs, also called as erythrocytes. So these cells, these are the most common type of blood cells. They are very much flexible because of their shape. We'll discuss it later. They do not have any organelles. They are non-nucleated cells, but they contain hemoglobin in the cytoplasm. And these cells, these are the primary means of delivering oxygen to the body tissues. The lifespan of RBCs, it is around 120 days, and these are degraded in the tissue macrophagic system of the body. Now, the size of a normal RBC, it ranges from 7.2 to 7.4 micron, whereas its thickness varies from center to the periphery. At the center, it is around 1 micron, whereas at the periphery, it becomes 2 micron. You can see the changes in the thickness and the overall size in this figure. We'll have a word about the shape of normal RBC. Normal shape of RBC, as you can see here also, it is of biconcave shape, which is maintained by the proteins in the cell membrane, RBC membrane. We'll discuss about proteins later. But this biconcave shape, it has certain advantages, like about the surface area to the volume ratio. This shape permits changes in the volume of the cell so that the cells can bear changes in the osmotic pressure and they become much resistant to hemolysis. Now, this particular shape permits the passage of these cells through narrow capillaries. Okay, so we shall discuss about the proteins that contribute to this biconcave shape that are present in the RBC membrane. So this RBC membrane, as you can see in this figure, it has a lipid bilayer. It has membrane proteins we had talked about. There can be presence of integral proteins like band 3, glycophorins, or the proteins that are present at the periphery called as peripheral proteins such as spectrin, ankyrin, actin. Okay. It has certain carbohydrate residues also. Now this RBC membrane, it accounts for the antigenic, mechanical and the transport related characteristics of these cells. Okay. Now about the composition of RBCs. Around 62.5% is water, 35% goes to hemoglobin and rest 2.5%. It includes glucose, lipids, proteins, enzymes like carbonic anhydrase, very important enzyme, derivatives of vitamins and ions like sodium ion, potassium ion, calcium, magnesium, phosphates, sulfates, etc. About the normal values, the values of RBCs, they vary in males, females at the time of birth and in children. As you can see here, the count is more in males as compared to females, and the RBC count is more at the time of birth as compared to children. There are several reasons to this. We'll discuss all the reasoning based question in different slides. Okay, but clinically 5 million per cubic mm RBC count, it is taken as 100%. Now about the variation in the size and shape of these cells. There are two terms. First is anisocytosis, in which there is change in the size of RBC. We had discussed that normal size of RBC, it ranges from 7.2 to 7.4 micron. So the term is normocyte for this. If the size becomes lesser than that, the term is microcyte. If the size becomes larger, the term is macrocyte. Micro means small, macro means larger. Another term is poikilocytosis. there is variation in the shape of these cells. Normally, we had discussed that the normal shape of RBC is biconcave. In case of sickle cell anemia, they become sickle shaped. Okay. In case of hereditary spherocytosis, they can become spherical in shape. Okay. In case of thalassemia, there is also change in the shape of the RBC. They can be fragmented also, say histiocyte. They can be in form of tear also, as you can see here. Okay, the cryocyte, stomatocyte, codocyte, etc. etc. Codocyte, these are the target cells seen in case of thalassemia and in case of chronic liver disease also. Now, about the variation in the number of these cells. There are two terms. First is polycythemia. Poly means more. When the RBC count it becomes more than 6 million cells per cubic mm, it is called as polycythemia. And as the cells, these blood cells, these are synthesized in the bone marrow. So for the increase in this number of cells, there could be increase in the bone marrow activity, or there could be increase in the production of erythropoietin. EPO, it stands for erythropoietin. Erythropoietin, it is basically glycoprotein. Okay, that is responsible for erythropoiesis and its regulation. So the increase in the number of these cells, it could either be physiological or pathological. Now there are certain terms to describe polycythemia. We can just have a word about them. One is primary proliferative polycythemia, in which there is tumor of the bone marrow and the RBC count, it can rise up to 7 to 8 million cells per cubic mm. If there is something like primary, there could be secondary also. Secondary to certain condition. It is a way to memorize. Secondary polycythemia, if a person resides at high altitude for a longer duration, in case of dehydration, shock, cigarette smoking, or some congenital cardiac diseases that can lead to hypoxia, cyanosis, or in case of chronic obstructive pulmonary disease, secondary polycythemia can be observed. Another term is relative or apparent polycythemia. Apparent means normally cell count is okay, it is normal, but there is decrease in the plasma volume. So the cell count appears to be high. In reality, it is not. In reality, what is there? There is decrease in the volume of plasma, but RBC count is normal. It is seen in case of dehydration. Now, opposite to polycythemia is anemia, in which there is decrease in the RBC count, and when it becomes less than 4 million cells per cubic mm, it is called as anemia. Now about the functions of these cells. As these cells, they contain hemoglobin, so we can say these are the primary means of gas exchange. They carry oxygen from lungs to the tissues and delivers carbon dioxide from tissues to the lungs for oxygenation. These cells, they contribute towards viscosity of the blood, and viscosity determines peripheral vascular resistance, and peripheral vascular resistance, it is a function of diastolic blood pressure. Third function is these cells, they play a very significant role in the regulation of acid-base balance inside the human body. Okay. Now that was all about the characteristic features, functions, normal count or variation, about number, size, shape of these cells. Thank you very much. Please keep on liking and subscribing the channel. In the next lecture, we'll be discussing about packed cell volume, ESR, red cell indices, osmotic fragility. Thank you very much.